Hello! Hello, everybody. It's week 13. How can that even be possible? Anyway, only 2 more weeks to go. Feel like I'm getting dragged through this semester. I don't know about you, but anyway, here we go. It's almost over. The light is shining at the end of the tunnel. And if you've been, on the ride, and it's rough, just keep hanging on. Like, I'm really just impressed and inspired and amazed that all of you are continuing to stay focused and get stuff done and just be in this game even with all the stuff that's going on in the world. So kudos to you. So for week 13, there's a lot of stuff, but it's not all stuff to do. So just take a breath. If you looked at it, you're like, ah! It's not all stuff to do. A lot of it is things like the CSI stuff is files that you want to refer back to. A little note about that referring back to piece. I just finally, sorry it took so long, got an assignment graded. I don't even remember right now what it was. Last night I finished it. That's my brain for you. And so many people are forgetting how to cite sources. I can't recall, but it was early on that there's a whole module, thing about a file in the module about how to cite sources. Please refer back to that. Don't forget that. I expect by now that you know how to do it. So I dock points if you don't, okay? Anyway, so there's a lot of stuff to talk about. So cell cycle and mitosis, this is like, 7th grade, 10th grade, high school biology stuff. But you may not be familiar with it. Prophase, metaphase, anaphase, telophase, the stages of mitosis, and the fact that cells go through these cycles. All of your cells, your brain cells, get stuck at one spot. That's why they're so precious. But you need to know about the cell cycle and then add on meiosis, which is how gametes are made. So we get germ cells, eggs and sperm, that have half of the complement of all of the DNA needed to make a human, so they can get together and make a whole person with copies from mom and dad of new combinations of genes that never existed before, because it includes genes combined in one nucleus with combinations of traits from your grandma and grandpa. Okay? Anyway, and how that whole cycle is regulated in places where there are stops, and what happens when that doesn't work, which is the introduction to cancer. So then, of course, soft chalk, just to go over that. And then we have the, so this is some of the ways that it's regulated. So again, we're looking at what can go wrong. Okay, so, angiogenesis, which I believe is adding more blood flow to an area, resist cell death, a whole bunch of information here that contribute to cancer. And usually it's more than one thing. So it's a useful thing to learn about, because we have so many issues, so many people having different types of cancer. There are so many different kinds of cancer. It's not all one disease. And it's just good to be aware of. It's about your health, but it's also about physiology, which is part of why we cover it in this class. And then a discussion post, the hallmarks of cancer. So what are some of the traits of cancer? Here's angiogenesis that I mentioned. There's only 4 groups, so I made the groups be 6 people so that each one of these things gets covered, one hopes. Because the idea for this is that you will need to be somewhat familiar with all of these articles. So rather than you reading all 5 of the articles, each one of you reads one and then you report back, right? That's how it's supposed to go. Now, I added 6 people in there because I don't want anybody to get skipped. I don't want, person number 5 not to be there. So if you are the 6th person in a wonderful group and everyone's already posted, then you get to choose whichever one you want. Okay, so it's a 2-post thing. First post, you read your article, write a short summary, an example from the soft chalk is very good. Cite them fully if you use outside sources. And then the second post, which you should be familiar with, is you respond to someone else's post. Yep, got it? Okay, there we go. And then a little quizling, and then the quiz for the whole section right there. And then we move right along. Now, this is only for 73132. Anyway, so we're going to talk about that. I think I made it easy and put a little tab up there at the end. So if you're in 73150, the other class, then you can stop when we talk about that. Let's see. I think I got all those pieces there. And then we move on to, because we were moving on to the next module. We'll talk about this at the end of this little screencast-o-matic. So for the CSI Lab, there's a whole bunch of content in here And this is stuff that you're going to need to refer back to. So there's like, how do you do hair analysis? I think I have that stuff up here. Open. Let's see. No, it's the same one. Okay. Sorry about that. I have it open again. Okay, so here we go. There's a little soft chalk with stuff. Of course, you go over that, right? Usually the soft chalks are pretty decent. Please note that you need to click through here. So we look at, oh, that's funny. Okay. We look at the hairs, right? And then their exemplars are examples of hairs, right? There's a bear hair. That was kind of fun to say that. Anyway, buffalo. Why would there be a buffalo hair in this? Anyway, camel. I'm sure there's a camel hair in the mystery. Just kidding, I doubt it. Anyway, so these are hairs to compare to. So the soft chalk teaches you what to look for, right? Like I think that's called the medulla in the middle is diagnostic to different types of animals, and even I think that's called the cortex on the outside. I'd have to look at the soft chalk again. But it's diagnostic, so you want to be clear about what this is telling you based on what the exemplars look like, right? You do, right? Don't you? You want to know a lot about hair. So hair is one of the things you need to be familiar with to try to solve the mystery of who killed Karen Vinson. Another piece of evidence is evidence from UV light testing. So body fluids glow under UV light. There's so many good, awful jokes I could make about that, but I will refrain because of time. And then you want to see, like, now what's glowing here? Now, blood is a body fluid, but it does not glow for whatever reason. It doesn't. So it actually looks super, super dark, like it's like a dark black, blacker than the surrounding area, but that's not diagnostic for blood. So if it doesn't glow, it doesn't mean that it's blood. It could be a bunch of things. So let's take a peek there at, here we go. So, oops, I don't think I did the hair analysis. Sorry, the UV light testing. I didn't. Anyway, so it shows you some pictures of different examples and then exemplars of what it looks like, different body fluids glowing with the UV light. Now, if you think there is blood in a sample, the diagnostic test, meaning it actually tells you if it's blood or not, is the Kastle-Meyer test. And this is a chemical test. And you have to go through a procedure and then look at your example. You have to have a positive control, which is blood, and a negative control, which is usually water, so that you know that your materials were fresh and that the test worked. Because if your blood sample doesn't turn out positive, then you've got something wrong. If your water sample turns out positive, you've done something wrong. So here we go. So here's the negative control. It looks as it should. And the positive control gets oxidized to this bright pink color. And then you have to look at these samples and explain. It can't just be like, oh, because it looks like that. Sorry, I'm not trying to make fun of anybody. But then you would explain, what makes that happen. You probably want to use the word hemoglobin and say a little bit about why this looks the way that it does. Yeah? I would want you to do that. That would be a good idea. Next, we have the DNA fingerprinting. So this goes back to stuff that we already learned about gel electrophoresis and how it chops up pieces of DNA. And the small pieces move further down the gel and the big pieces are stuck back towards the beginning. And then we look at evidence from Karen Vinson's case. Is it here? It is. Okay, and here we go. Here are the lanes. So standard is just like a way to, it's like a control to make sure that you did it right. Here we've got Karen, the victim right here. Patrick, right? So you just go down and you match the lane to the person or the piece of evidence. And you explain what this information, given this DNA, tells you about the case. And when you write that up in the evidence analysis, the last part of this module, you want to be really clear that you're not just saying, oh, because it matches. Well, what matches? Why did that Kastle-Meyer test show positive? Because there was hemoglobin on that sample, which comes from blood, and that's why, blood, hemoglobin with these compounds or whatever oxidizes to bright pink. So show me that you understand the chemistry and the biology of what's happening. This is not like a board game. It's not like Clue, and you're playing matching. Like, you want to show that you've actually made the link between what we've covered in class about DNA and how the enzyme chops it up, gel electrophoresis, and the content that we've covered in class. Clear? So here we go. Evidence collected, refer back to these things if this is not clear to you. And then, species. So this part right here, answering these questions, is what gives you the bits and pieces that you're going to need for later on. So I keep stressing this, but for some people it's either just too much going on in the world or just not paying attention. This class is designed to help you succeed because each assignment on projects builds from the earlier assignments. So if you try out right here and you're like, oh, I don't understand it because you didn't do all these other steps, you're giving yourself a huge amount of hassle and headache because you didn't do the build-up pieces. So I behoove you to go back, start at the beginning like this one right here. Make sure you do the beginning steps well. It's like building a castle out of sand or whatever, right? Like Jimi Hendrix. Anyway, don't build a house without a strong foundation. Don't try to do the project at the end without all the pieces that built up to it. Okay? Now, I'm done with that. I'm going back to this for those of you in 73132. Okay. Now, just so you know, 73132, you don't have extra work. We did chop out something in week 14 that you don't have to do that the other guys do because you have this. One hopes it will rain because ideally this happens after the rain. So you are going to make a little public service announcement about cleaning up our neighborhoods and what happens to the trash in our neighborhoods. So a second trash survey, ideally it would be after it rained, right? So you go out and you do the same thing you did the first time, except after it rains. And again, we're hoping that it rains because that's really the point of it. But even if it doesn't, we're still, the assignment still, it's a good one. Write a blog post, so it's short, it's less than 300 words, but it should be short and snappy. Like a blog post is meant to get your attention. It's meant to communicate to like somebody walking on the street or somebody at the grocery store or like, just catches your eye when you're scrolling online or whatever. That, actions, where is it? Yes. Action steps. What is the impact of having trash in your neighborhood? And what difference could it make if that trash wasn't there? Right? So, anyway, tips to do that. Straightforward, interesting, personal, first person. And then some examples, which is really useful stuff. So this is like getting the word out, a little bit of activism, which is really, I think, a really important piece of education, applying what you've learned. And then this is not due this week, by the way. So again, if it rains this week, I'm hoping it rains for my garden too, but you may want to do it now because ideally this happens after it rains, or until next week when it's due. If it doesn't rain at all, then you just got to get it done. Clear? I hope that's clear. Okay, home stretch, guys. Keep up the good work. Bye.